Before we jump into the cycle itself, let's remember one important thing. A molecule of glucose first goes through glycolysis in the cytoplasm, yes, where it's split into two molecules of pyruvate. These pyruvate molecules then enter the mitochondria and are converted into Acetyl-CoA. Yes? Think of Acetyl-CoA as the ticket that allows carbon atoms to enter the cell's main energy factory. Now the real magic begins. The cycle starts when Acetyl-CoA carrying two carbon atoms, yes, combines with oxaloacetate, a 4-carbon molecule, yes. This reaction is catalyzed by the enzyme citrate synthase, yes. Citrate synthase. Got it? Together, they form citrate, a 6-carbon molecule, yes. Notice something interesting. Oxaloacetate isn't consumed forever. It's like the starting position in a relay race. It disappears temporarily but returns at the end of every lap. So step 2, isomerization, yes? Next, citrate is rearranged into isocitrate, yes? This reaction is carried out by a kinetase. No energy is produced here. The molecule is simply reshaped so it becomes chemically ready for oxidation. Step 3. Now the real energy extractions begins. Isocitrate is oxidized by isocitrate dehydrogenase. Yes? One carbon atom leaves the cycle as carbon dioxide. Yes? Here. At the same time, NAD positive captures high energy electrons and become NADH. Yes? This is the first major energy carrier produced by the cell. Yes? First major energy carrier. Step 4, second oxidation. Yes. Next, α-Ketoglutarate undergoes another oxidative decarboxylation. Yes. Again, another carbon is released as a carbon dioxide. Yes. Another NADH molecule is produced. Second NADH. The remaining 4 carbon molecule becomes succinyl-CoA. Yes, succinyl-CoA have 4 carbon. At this point, both carbon atoms that originally entered with acetyl-CoA have now been released as CO2. However, not necessarily during the very first turn of the cycle. A fascinating detail explained in Campbell Biology. Step 5. Now comes one of the few moments where the Krebs cycle directly produces usable cellular energy. Succinyl-CoA is converted into succinate. Yes? The energy stored in its high energy bond is used to produce one molecule of GTP or ATP. In some organisms, this process is called substrate level phosphorylation. Yes? Substrate level phosphorylation. Yes? But this process did with this enzyme, succinyl-CoA synthetase. Yes? Now step 6. Succinate is now oxidized into fumarate. Instead of producing NADH, this step generates FADH₂. Yes? Here's something unique. Succinate dehydrogenase is the only Krebs cycle enzyme embedded directly in the inner mitochondrial membrane. It's also known as complex 2 of the electron transport chain. That means it participates in both the Krebs cycle and oxidative phosphorylation. Next, water is added to fumarate by the enzyme fumarase. Yes? This forms malate. Yes? Again, no ATP is produced here. The goal is simply to prepare the molecule for the final oxidation. Now, step 8. Finally, malate oxidized back into oxaloacetate by malate dehydrogenase. Yes? This enzyme is malate dehydrogenase. Yes? Another NADH molecule is produced here, the third one. Yes? Now we've returned to oxaloacetate. Yes? Exactly where the cycle started. The engine is ready for another Acetyl-CoA to enter. So, what did one turn of the Krebs cycle actually accomplish? Yes, I summarize for you. From one Acetyl-CoA, the cycle produces what? This 3 NADH, 1 FADH₂, 1 ATP or GTP, 2 CO2 or carbon dioxide. Have a good time.